to be able to say that up here. That was at University of Wisconsin-Madison. Come up. And yes. I have a little bit more to go. And now he is the Neufeld Chair of Anesthetics at the University of Sydney and Royal Prince Alfred Hospital in Sydney, Australia. His research has given us important insights into the pathogenesis of delirium, spanning two broad themes. One is the mechanisms and consequences of delirium, and two, the mechanisms of anesthesia. He's currently the recipient of 2 R01s, along with other NIH funding, collaborates broadly in the US, which he'll talk about, and is going to share his incredible work with us today.
So thank you very much to Heidi and ADS for the kind invitation to come and speak. I was a bit late this morning. I wasn't confused, but nor was it intentional. There's another backstory there. But thank you for taking over with me. And I loved the last talk. My very first research endeavors 20 years ago were in neonatal pain and how anesthetics might modulate that in mice. And that basic science foundation has driven a kind of a passion to understand the pathophysiology of events that occur in clinical care, and delirium is obviously a key passion as well, to understand what is the underlying substrate of events that occurs in a patient's brain when they're experiencing this mixed symptomatology, which is delirium, this syndrome that we try and group together. I'd like to start with the traditional in Australia. So I moved to Australia in 2020. I'm a Brit. I spent 5.5 years in Wisconsin and now at the University of Sydney. And it's traditional when giving a talk at the University of Sydney to acknowledge the tradition of custodianship in Lord Country in which the University of Sydney stands, and we pay respects to those who've cared and continue to care for country. And it's part of the reconciliation we have as an Anglo-Saxon who, when we, for want of a better phrase, colonised Australia, we didn't necessarily respect the indigenous population where it should be. So that's part of the work which we're doing, and part of the learning that I have. Other things to say, I don't have any conflicts of interest. I'm desperate for some, if anyone's interested. Please come and see me afterwards. I'm very that sort of thing. Do I have any biases? Yes. So the topic, Heidi kind of said, would you come speak, what sort of thing would you like to talk about? I said, well, wouldn't it be great if someone could give a talk on pathophysiology and will it lead to new therapies? And Heidi said, well, why isn't that you? And I thought, another good idea that I didn't really mean to let out of my mouth. So I have a big bias, this is a pitch, I suppose, for funding, but it's a pitch for to take you all on the journey which we're going on as a group, and many of you are already on. So I do have a bias that I do believe that understanding the pathophysiology of delirium is going to yield new therapies. I've managed to persuade NIH that that's a good idea so far, and we'll see if I can continue to do so. And I'll see if you're convinced at the end. If you haven't read this paper and you're interested in the pathophysiology of delirium, and I hope you all are, I really recommend you do read this paper. I'm not an author, so this is one of the few parts of this talk that doesn't feed my megalomania. But please look up this work, and there's some really important explanatory diagrams within this to help us understand the pathophysiology of delirium. A key idea that's been around for about 70, 80 years has been that delirium results from cerebral metabolic insufficiency. So there's an energetic failure in the brain. And essentially, mitochondrial failure, breakdown of the TCA cycle, loss of oxidative phosphorylation means that there's an energetic failure. We're not converting glucose and oxygen into energy in terms of ATP. Now the correlates for this idea are somewhat sparse, but an important one has been the measurement of CSF lactate, which I'm going to come back to at the end because I'm not sure CSF lactate itself is a biomarker for energetic failure, and that's something which I think there needs to be more research on. Nonetheless, it does stand to reason that you can reproduce the symptoms of delirium with hypoxia and hypoglycemia. So it is a very logical conclusion that there is energetic failure in the brain, and that's a key underlying feature of delirium. complementary, and all of these models overlap, to be honest, is the neuroinflammatory model with breakdown of the blood-brain barrier. And this wonderful diagram, again from that review by Wilson, really describes how systemic inflammation in the blood leads to activation of white cells, breakdown of the blood-brain barrier, infiltration of white cells into the central nervous system, further elaboration of a central inflammatory response, activation of microglia, which leads to cytokine release, neuronal dysfunction, and neuronal injury. And astrocytes, which are really important for the energetics, we saw them on the last slide. Astrocyte failure means that they're no longer able to supply neurons with the energetic supply they need in terms of lactate and pyruvate. It means that there is also a cerebral metabolic insufficiency. And there's quite a lot of data which supports the idea that central inflammation is a key feature of delirium, and seminal work by many people in the room have shown that CSF cytokines are associated with delirium, so you can measure inflammation in that central compartment, and it's associated with delirium. And so we start to build a picture of, well, how are we going to optimize energetics, how are we going to dampen cerebral inflammation, how are we going to protect the brain. The brain is this immune privileged organ that has this wonderful blood-brain barrier that's great until it's not working. And then suddenly we've got dysfunction of that and an elaboration of a central inflammatory response that seems to lead to delirium. And again, in the Wilson review, they try to put this together nicely in this diagram showing the brain where those arousal nuclei at the bottom there, including the basal forebrain, the ceruleus, serotonergic nuclei like the raphe, dysfunction leads to changes in arousal, which we commonly see in delirium, activation of microglia, central inflammatory response, neuronal injury. And I think, and that's where the talk is going to shift, it's important to think about the communication within the brain and this common final pathway where there is failure. I don't know how to use this, I can't see the screen. I was going to point at something. Can you see the cursor? Maybe if I just like wave. So if you look at the bottom right of the screen, as you look at it, there's a green blob that says delirium. Right? Just above the green blob that says network dysfunction, and that's where we're going with this, because all of these associations are really interesting. We've observed them in patients, but here's the question. How do these changes lead to delirium symptoms? Without understanding the brain, which is a big topic, it's going to be hard for us to really understand how the brain works. So we need to break down these very simple models of the brain initially. Undoubtedly what I'm going to propose is wrong, because there's no way that I can use a simplistic model to explain this. We need to generate tractable hypotheses that we can test and correct. But I'm going to suggest a model for that network dysfunction that we can test in studies. We can prove it's wrong, we can learn in that process how to make a better model of what's happening. The symptoms of delirium, which is why I think we're all gathered here today, because it's that the symptoms that the patient experiences at the bedside that's distressing for the patient. We also need to understand what that link is to later problems like cognitive decline and dementia, and how we can prevent those. So our working model is that delirium is a state of cognitive disintegration. This is a working model that used to be less than a decade old, but now about 12 years old, about how we can fit models for how the brain understanding of delirium. And we're calling it the cognitive disintegration model because it's easier than saying the title for the paper. But what the critical features of this model is that the brain is made up of modules which all have a specialized function. The brain works because those specialized modules interact. So you have areas in different areas of the brain which have specific function. For example, at the back of the brain you have the parietal area here that's highlighted, and at the front you have frontal cortex. Together they call something, so they form something called the executive control network, which is really important for reasoning. But parietal lobe is much more important for spatial navigation, understanding where you should be in space, than the frontal lobe. The frontal lobe has this higher order function of reasoning, trying to deduce where you should be in space. They work together, they form a synthetic, they integrate information. If the brain is not allowed to integrate information across these specialized nodes, then the brain is not functioning in the way that it's supposed to. An alternative thought process is, if you're looking at me now, you'll see that I'm wearing a blue suit, but you don't experience the shape of my suit separate from the color. Your brain has color neurons, it's got neurons which respond to the shape of objects, it integrates information across those two to give you a perception of me. Our thinking in delirium is that there's fragmentation of this function, so that people get disjointed understanding of their environment, of where they are in time, of people. And there's simpler functions which also become disabled, like attention and arousal. And we think that there's some critical mechanisms that can link this fragmentation of networks to some of those earlier pathophysiological observations around energetics and inflammation. And one of the critical links, we think, is cortical slowing. So this is a description. You can measure the EEG definitely. We can measure this in the EEG, and it's a description of how synapses slow, they become more synchronized, and they become less able to process information. Now, it's been known for 70 or 80 years, EEG slows in delirium. Romano and Engel really deserve all the credit in this space. And they also were early pioneers of the cerebral metabolic insufficiency theory. And so credit where's credit due, this is amazing work. So I think, if I remember right, this is from 1956, this paper. And it shows at the bottom, a patient with mild delirium, moderate delirium in the middle, marked delirium at the top. And you can see that the wiggly line changes as you go up, and it becomes slower, so that you see a fast wiggly line at the bottom, and it's going progressively slower, that wiggly line. And that slowing represents synchronization of postsynaptic potentials in the cortical regions underlying the electrode. And the question is, can we reproduce this? These data showing that this slowing occurs proportionately to the symptoms of delirium. So a key feature of the work I'm trying to do is understand, because it's hard to show causality, but at least if there's proportional changes between the physiological variables we measure and the patient's symptoms, we're heading in the right direction. So this is some work from Sean Stanley, who is now in Montreal doing a PhD, and then a couple of grad students, Rosalina Manti, Heidi Lindroth, if anyone knows Heidi, and Cameron Casey. And these students are working so closely together to pull off what was a big project. I'm not going to try and describe all the data in this paper, because it was a big humor. But one of the critical things we wanted to do is reproduce Engel and Romano's discovery in the perioperative setting. We wanted to take patients and do an EEG before surgery and an EEG after surgery. And these are just two examples of patients. One who did not get delirium after surgery, and one that did get delirium after surgery. We'll talk about the pre-op data. So this is when they are before surgery, no one's got delirium at this point. And you can see that there's this nice, tight, fast alpha rhythm that's really evident in the EEG. The patient who goes on to get delirium actually has a much bigger alpha oscillation. This was unanticipated, and I don't have a good explanation for you about that. There's more in the paper. If you're intrigued by that, well, please come and talk to me afterwards. But that's not the most important finding on this slide. If you look at the bottom right, you'll see the patient who gets delirious, that fast rhythm is completely fragmented, and you've got this big slow oscillation that's appeared. Actually, in the patient who didn't get delirious, there is a bit of a slow oscillation at one point in their EEG. I would point to it if I could point to it. And that's also important because after surgery, just because you don't reach this dichotomous threshold for delirium doesn't mean you're not having inattention or other problems, arousal problems. So this is an important thing that we were talking about it yesterday in a NIDIS meeting. Richard Jones taught me a new word, dichotomania. One of the things which we're trying to get away from is this binary construct of delirium, because we think that there's a biological continuum, that there's not many things in biology that's a true on-off switch, and we think that delirium is a continuum of severity of symptoms. That's unplanned, that music, not part of the talk. I don't know the rest of the lyrics, sorry. So we wanted to see whether the EEG slow really does slow, and that really does correlate with delirium severity. And this gets back to this idea of inflammation too. So on the bottom left is a graph. What we did was we had a 256 electrode EEG against a cat, but for the purposes of the description of the data, we're going to look at one electrode on the occiput at the back of the brain. On the x-axis, on the first graph on the left, you've got delirium severity and how much it changed. We did that with the delirium rating scale. Was the work we Heidi probably did most of that with the delirium rating scale. And then on the y-axis, we have slow wave activity and how much that's changed from pre-op to post-op. And you can see there's a fairly strong correlation between how much the EEG has changed pre-op to post-op with the accumulation of delirium symptoms. So we think that cortical slowing is really important, or there's an association anyway, I should say, between the cortical slowing and delirium severity. We then looked at inflammatory cytokines. These were assayed in plasma, based on the idea that systemic inflammation is a key driver of central inflammation, and the central inflammatory changes are associated with these changes in the cortex, in the synapse, which we can observe in the EEG. And you can see there's a correlation between the anti-inflammatory cytokine IL-10, how much that changed with slow wave activity, and similarly for monocyte chemotrypsin protein 1 or MCP1, which is a pro-inflammatory cytokine that's really important for driving monocytes to kind of come into the brain through that broken down blood-brain barrier. So inflammation can drive these EEG changes, and we've shown these EEG changes are associated with delirium symptoms. What about integrating in the brain? So I said the brain is this modular structure, and the way that it functions, it needs to exchange information between these different modules. So how can we link the different modules with the EEG data? And the way that we did that was we calculated something called phase lag index. So you have two signals which are going up and down together. If they're going up and down together, the phase lag index is high. And if they're doing their own thing, the music's supposed to stop. If they're doing their own thing, then they're not correlated, okay? So in this situation, there's not connectivity, and when they're working together, there's high connectivity. And we calculated the change in the phase lag index from the y-axis of these graphs on the right. And on the x-axis, we have delirium severity, MCP1, that pro-inflammatory cytokine, IL-10, the bottom left, which is the anti-inflammatory cytokine, and slow wave activity. And you can see there's a negative correlation with all those variables with connectivity. So that delirium symptoms correlated with connectivity in the EEG, which is the exchange of information between these two modular areas of the brain. Pro-inflammatory cytokines and anti-inflammatory cytokines, which rise in compensation for those pro-inflammatory cytokines, also are associated with connectivity change. And there's a strong correlation with how much the EEG changes to drive that slowing in the brain. And we think that the reason that slowing in the brain is key is that the brain enters this firing pattern where rather than neurons able to sort of interact as they need to, they get
forced into something called bistable firing. The whole caudal column is on or it's off. It's on or it's off, and you get the big slow wave, oh no, and then it can't process information in the correct way, and it can't. Those modules can't work together to process information and exchange information to bind what you're seeing in your environment, creating a synthetic you can interact with. Now, how long are we going for? Half an hour, 3 hours? So this is a bit of a plug now for basic research. Well, this is all very well, but there are so many things changing in the perioperative period. These patients have pain, they have analgesic drugs, they've had different anesthetic drugs, they've lost a lot of bloods, haven't lost any bloods, they've been given blood. There are so many different things that change, and it's all very well that we've pinpointed these points in time, measured the physiology, and we've said all these things are changing together, but these could all just be epiphenomena of something that's really much more important. So to try and get at this, we turned to a basic animal model where we took mice and we gave them a vehicle, saline, or inflammation, something called lipopolysaccharide, which is a gram-negative endotoxin that drives a big inflammatory response in the mice. And this is work led by Ziad Sultan, and Matt Banks is the PI. He is a collaborator for UW-Madison. And what we were interested to see is, can we reproduce these EEG changes which we see in the human, in the complex state of delirium, in a very reductionist way, with inflammation in a mouse model. So the short story is we can, and these are 2 EEG traces from a mouse treated with a vehicle before and after they get the injection on the top, and the lipopolysaccharide before and after. And before, there's not much difference in the EEG, they're both pretty active. If you look after, there's a bigger slow wave activity in the low LPS group. And this is in LPS terms, in mouse experiments, most people studying this think we're mad giving such low doses as this. We call it low LPS. It's only 25 mics per kilo. Most studies will give milligram doses. And the reason we gave such a low dose is we want the mice to not be so sick they can't move around. It's just movement changes the EEG. There's so many things that change the EEG. So we wanted to limit those confounds. And unfortunately for me, Matt Banks is a really, really good scientist and a stickler. And so this took us years to do so we could get as many control as right as possible. But what we found was that if you look at the bottom graphs, that there's a dose-dependent effect of inflammation on slow wave activities. The dose of LPS, when we gave 25 or 125 mics per kilo, caused a differential effect on slow wave activity. So how much inflammation you get is proportional to how much the brain slows. And a breakdown in the connectivity, which is that alpha WPI, that weighted phase lag index. So, and the reason we've got different time points is we did a time course to make sure that there were transient effects, that there was prolonged effects. And it's possibly easier to see on this graph, which is now another correlation plot looking at the inflammatory changes measured by interleukin 6 and MCP1 on the x-axis, and how much the EEG slowed on the y-axis. You can see there's a pretty strong correlation. The different shapes of those circles represent different doses of LPS, but a strong correlation between how much inflammation there is in the brain and how much the EEG slows. The sharpest amongst you will have noticed there are some red dots. Now the red dots are really interesting because inspired by work by Colm Cunningham, who'd done many, many experiments prior to us doing this, like in animals which have relevance to their pathophysiology, and I'm a big fan of Colm's work. He'd shown that piroxicam, a non-specific non-steroidal anti-inflammatory drug, could reverse the cognitive changes of lipopolysaccharide. And Cliff Saper, who's a neurologist from Harvard, who previously proposed that prostaglandins were key in sickness behavior and could have a link in delirium pathophysiology. So we gave piroxicam to the mice, and what we showed was that we could change the EEG slowing, but we didn't change the cytokine. The brain inflammation was still happening, but the prostaglandins, which were downstream of that inflammation, were no longer driving an EEG slowing. So that paralleled Colm's work with cognition. I think it gives us some confidence the EEG is a good translational biomarker for us to use to study delirium pathophysiology. And it really showed that prostaglandins are an important link drives adenosine release in the brain. And we were able to then go on and show that if you give the mice caffeine, you could reverse the EEG slowing. Therapeutically, I don't want us all to go out of this room and start giving everyone NSAIDs, right? Because they're our most vulnerable patients. They get vulnerable to the side effects of patients with delirium. I don't want people to have GI bleeds and renal failure and those sorts of things that we should consider, but just I don't want that to be some sort of default thing. I do think that every ward should have a barista able to make espresso coffees. Briefly, I'll mention this. Please don't take photos of this or tweet it because partially I'm not going to explain it in full, and partially because it's not out there, but it's going to be coming out, I hope, or going to be submitted very soon. But we wanted to understand the brain a little bit more fully than we have done with just looking at scalp EEG responses. And we've done something called dynamic causal modeling, which is a way of looking at the cortical hierarchy in more specifics, arguably within the confines of a model. And what it does is we create a model of what's happening to an auditory evoked response and how it's transmitted through the brain from primary auditory regions up to superior temporal gyrus, more complex higher order region for auditory processing to inferior frontal gyrus. So there's a hierarchy that goes from temporal up to frontal cortex and back. And we put an input to the model, which is an auditory evoked response. We're measuring the EEG activity on the scalp, but then we're modeling what's actually happening in those brain nodes. And one of the things that's really key that seems to be happening is that the information is going up in the brain, but there's increased inhibition happening at this superior temporal gyrus region. So it's not in the lowest level of cortex, not in the primary auditory regions, but are in higher order regions, which then stops the information processing and exchange of information in the hierarchy. There's more to come on this, but this does suggest that there, at least in this group of patients, that there may be therapies we can think about that are targeted to that inhibition that's happening in the brain. And what was really interesting as well is the feedback projections, which seem to be most correlated with delirium severity and that inflammatory burden. And those feedback projections will be important because if the information is going up, say it's even being processed, the important thing is it's fed back down the hierarchy. So when you're seeing me, and I'm standing by the podium, if you were to close your eyes, everyone close their eyes. Checking. Right, open your eyes. So the reason you laughed is you were expecting to see me at the podium, right? Your brain had a prior expectation of where I would be. And when you opened your eyes, I was no longer there. And the reason that you have that prior expectation is that the information has been fed back down the cortical hierarchy. And your primary auditory region, or your primary visual region, this was expecting to see me. And then you've got a big error signal like, hold on, he's a lunatic. And the important thing to realize is that it's that loss of feedback connection which we think is important here, it's important for the humour which we've had, but it could also be important in delirium. Right? If you're not able to update your model of your environment, you're completely disorientated. Like, where are you? You're in a hospital. Where am I? You're in a hospital. You know, it's this sort of the disorientation and it's loss of these specific brain functions which we potentially can model. I think are going to lead to new therapies. Now, I'm taking much longer to get to this point than I hoped, so I'll go very, very fast and skip a bit as we finish. So, just based on these few studies, I think there are a few therapeutic avenues for us to consider. What about targeting neural activity? What about targeting prostaglandins? I'm not the first person to say, Cunningham, I've been saying this for nearly 20 years. Can we modulate that? Is there a barista required in every ward? How are we going to target inflammation? At least in the post-operative period, that seems key. Do we have to be very specific? Are we going after one cytokine? Are we going after multiple targets? How are we going to design the studies to test that? Are these findings generalizable? Can we do them in the ICU? Can we do them in medical patients in delirium? And what about the patients who have dementia? Are we going to have the same, can we use the same therapies in those patients, or is the pathophysiology actually very different? And I think building from a conceptual model for how the brain works is a good way for us to start constructing that, because there are really good data on the conceptual models for how to dementia is happening and what happens to connectivity in dementia. So briefly, I'm just going to, if I have time, to talk about neuronal injury, brain injury in delirium, because I think this ties back to cerebral metabolic insufficiency, and I think there's important work to be done in this space. So firstly, a plug for the Australasian Delirium Association. We just had this pathophysiology meeting a week ago, which is a great trial run for this talk. And there are some important people that I'm going to point out. Glenda Halliday on the far right of the screen, she's a formidable dementia scientist who came to educate us about dementia. Next to Glenda is Reid Watner, who's the president of EDA. He's done some incredible work with CSF biomarkers, and it was great to know from you. Monique, next to Liv, is an up-and-coming EEG scientist, delirium research. On my right is Gideon Kaplan, who has done great work with CSF, working in some incredible neuroimaging. And some of the ideas we have here were what we've bandied about today. What you'll notice is I'm wearing my delirium presentation suit. I didn't want to challenge too many priors, so I wanted to wear exactly the same clothes. So, Cameron, Casey, and Heidi and Rosaline, you get to hear the team. So they did some great work, which we published in Brain in 2020, looking at the association of neurofilament light and axonal protein, and the association between that axonal protein as a marker of neuronal injury in delirium. We wanted to understand, are there dynamic changes in neuronal injury that occur in the setting of delirium? And really, we want to try and get at, are those driven by inflammatory processes or alternative processes? And so in a nutshell, this is what we found. In the top left graph, you can see non-delirious subjects plotted in blue and delirious subjects plotted in red as a group. And on the y-axis is the change in neurofilament light from pre-op to post-op data. You can see in the delirious patients, there was an increase in neurofilament light relative to non-delirious subjects. Next to that is the time course of the delirium changes over time. So you've got baseline, which is pre-op, post-operative day 1, 2, 3, 4 on the x-axis. On the y-axis, you've got the change in neurofilament light normalized to baseline. So everything's normalized to an individual's baseline, and you can see that it rises. Neurofilament light rises in both groups, but it rises more in delirious subjects. And I should shout out Liz Everett at this point, because Liz had shown neurofilament light changes perioperatively prior to us. We were able to then build on that with looking at delirium. On the bottom left, you can see a graph of delirium severity over time with the delirium rating scale, and you can see that obviously delirium severity is higher in the delirious subjects, so hopefully you can believe in that. Bottom right is the correlation between change in NFL on the x-axis and the peak delirium severity on the y-axis in the perioperative period, and there's a proportional change in how much neurofilament light was detected in the plasma and how severely delirious an individual was. And it's bizarrely linear for a biological process. Maybe with more data we'll understand the exact dynamics of that. But fascinating that we saw a proportional change. Neurofilament light actually also correlates with inflammatory cytokines, so we think that inflammation may be a key pathological driver of this axonal injury that we see, we think we're observing. Interestingly, when we put NFL and interleukin-8, which was the cytokine most strongly associated with delirium severity, we put them both in a model to predict delirium severity. They both independently predicted delirium severity. So what that means for me is that while there's collinearity between these inflammatory processes and these neuronal injury processes, inflammation, at least measured with just that one cytokine, doesn't explain all of the neuronal injury and its association with delirium severity. Now, in parallel, the neurovision study was happening, which was a study of about 1,000 patients in the perioperative period, and they had post-operative MRI scanning to detect covert strokes, which are these tiny lesions that you can see on MRI that don't necessarily have a bedside correlate that you can see with a neurological exam. These events seem to be thromboembolic and associated with injury, and importantly, they were associated with the incidence of delirium we wanted to do a little bit more digging, and this is work by Jen Taylor, who's a postdoc of mine who did these analyses in Sydney, looking at the patients that we recruited at the University of Wisconsin, who were both in a neurovision study and also in our local study, where we had blood biomarkers for neurofilament light. And what we found was that looking at the patients who had ischemic brain injury or not, so if you have ischemic brain injury, the S in red, there was a greater rise in neurofilament light in those patients. So there is definitely an association between a cerebral metabolic insufficiency, as you would say, from the thromboembolic process here, and the neurofilament light change. And I think it really drives a need to understand these dual parallel pathophysiological processes and show that we need to understand in an individual patient of these two potential pathophysiological processes which should we be targeting to benefit the patient that's in front of you. So we're going to need more than just one biomarker to explain how we're going to treat our patients. And of course, just to reiterate, this is an incredibly simplistic view of a complex disorder. It's undoubtedly going to be even more complicated than two biomarkers. We also have done some work looking at how tau changes in the plasma. And tau, I think, is a particularly interesting molecule. So just to shout out a few people, Tyler Hallway did this work, and Rick Lennox, as the penultimate author, is someone who's taken over in the Wisconsin study. He's a phenomenal scientist, has been really driving things forward. And what we did was we took our plasma samples and we sent them to Henrik Zetterberg and Kai Bernau, who are very generous, world-leading scientists from Sweden, and they ran our assays for total tau as well as they did for NFL. And they also ran it for really acidic protein, or GFAT. GFAT wasn't very interesting, it didn't show an association with delirium, that's the top graph, I'm not going to dwell on that, but tau was really interesting. Look at the time course here, divided by whether you have delirium or not. You can see that tau goes up in the delirious patients. It actually rises proportionally to delirium severity. But the critical thing about tau that's different to neurofilament light, and in fact different to all the cytokines we've studied, is that the recovery of tau predicts the recovery of delirium symptoms. Now, total tau, as it's referred, is a really interesting biomarker. It was one of the early biomarkers that was pioneered in dementia. But it kind of lacks a bit of specificity as to what it is, what it actually measures. And one of the things that's really curious to me is that with synaptic activity, tau is released in the synapse. So this could be a marker of either synaptic activity, there's more activity at synapses. I don't think that's true because EEG is being slower. That would not be compatible with more synaptic activity. It's kind of more likely to be associated with less synaptic activity. It's not increased firing, what is it? It could be synaptic pruning. And that's the research area which Rick Lennox is leading in a really interesting idea about how we can understand what is happening in those synapses and should we be preventing those synapses being pruned if they are being pruned. So I think tau is a really interesting biomarker, particularly because it's dynamics, particularly its recovery dynamic, where it recovers with the recovery in concert with the recovery of delirium severity. And just to reiterate, these are all associations. Causality is yet to be shown. So what about neuronal injury? Well, we've heard of the covert strokes associated with delirium. How can we prevent those? And the vascular hemodynamic arrhythmia events that we should be preventing to stop that cerebral metabolic insufficiency occurring. Again, should we be intervening with inflammation? What about tau? Do we need to have tau-specific interventions, or is this a marker of some other neurophysiological event that we need to understand better? And then finally, the blood-brain barrier, and there's a whole session on blood-brain barrier, so I just decided to put one slide or two slides in on this, and then if you're really interested, please come to the session later that Niccolo Turando, who's another great collaborator of mine, is running later today. In fact, Niccolo and I were in the lab in London 20 years ago, and he's an Italian. We were lab together 20 years ago, working in the same lab, and then he's an Italian who's now at Duke, and I'm a Brit now in Sydney, so it's funny how our paths diverge and converge over time. So this is Jen Taylor. These are the analyses she's done. Again, Rick Leonard's pioneering, helping push forward this work. And this is all I'm going to present from this study, because I do want you to come to our session later. But this is the pathophysiological model which we have for what's happening in delirium, and it's certainly complementary with some of Nicolo's work. That anesthesia and surgery is driving this peripheral inflammatory response, which helps cause blood-brain barrier permeability in the central inflammatory response. We measured CSF IL-6 to make that point in this paper before and after surgery, which was quite a Herculean effort. And then we get this CSF lactate response, which we also measured. And one of the things we think is really interesting about the CSF lactate response is that lactate is actually released in multiple settings by white cells as an anti-inflammatory modulator. And this was discovered with tumours, that white cells invading release lactate, and it would prevent the inflammatory response, the natural killer cell response, for example, to kill a tumour. So it could be that lactate is not a marker of cerebral metabolic insufficiency or anaerobic metabolism as we think of it in most terms, but actually is an anti-inflammatory effector that can activate something called the HCA2 receptor and lead to a subsequent neuroprotective response. It's also important for cerebral energetics. Lactate is an important supply of neuronal energy. So there are multiple other reasons why CSF lactate could rise, and I think there's a huge amount of work that could be done there in delirium pathophysiology that could be done in basic science and clinical science, and will be really informative for the future. So that's a plug for anyone who's desperate to find a new outlet for their energy. But finally, lactate also drives this prostaglandin D2, which we think is really important for slowing and would fit with us for being able to reverse the slowing with a non-steroidal anti-inflammatory drug. So we are getting some convergent pathways. I do think inflammation is key. I suspect cerebral metabolic insufficiency is also key. And understanding the balance in your particular patient is where I think that the future of delirium pathophysiology and tailored therapies are going to go. So with that, I'm just going to put in a few final plugs, which even though I'm over time. So we need to transition to hypothesis-driven clinical trials as soon as possible. We need to be understanding pathophysiology as part of those clinical trials, including these sort of biomarker studies. We need to fund basic goals that are really important to help ensure our understanding of what's happening in the clinical environment. We need to fund parallel translation experiments in humans so we can be sure that those basic models are relevant. And we need to study parallel disorders. There's so much happening in the dementia literature right now. I can't keep up with it. There's so much that we can learn from studying those. So finish by thanking funders, NIH, MRC, IRS, hopefully NHMRC. Well, I think I've been funded by NHMRC. And finally, the people who've worked for me, particularly Heidi, Sean, Cameron, Ziad, Jen, Jordan, and collaborators Matt Banks, Bob Pearson, Rick Lennox, who's now leading the Wisconsin efforts. Thank you very much.